Good morning, students. Myself, Dr. Manoj Kumar Pandey, Department of Zoology, Ganga Singh College, Chhapra. Today we will discuss enzymes. So, first of all, general features of enzymes. An enzyme is a biocatalyst that increases the rate of chemical reaction without itself being changed in the overall process. Virtually all cellular reactions or processes are mediated by enzymes. Enzymes have several properties that make them unique. As for example, most but not all enzymes are proteins. Enzymes are highly specific. Enzymes exhibit enormous catalytic power. It increases the rate of a reaction by lowering the activation energy. It changes only the rate at which equilibrium is achieved. It has no effect on the position of the equilibrium. Enzymes can be divided into two general classes: simple enzymes, which consist entirely of amino acids, and conjugated enzymes. Conjugated enzymes contains a non-protein group called a cofactor, which is required for biological activity. Removal of cofactor from a conjugated enzymes leaves only protein component called an apoenzyme, which generally is biologically inactive. The complete biologically active conjugated enzyme, simple enzyme plus cofactor, is called holoenzyme. A cofactor can be linked to the protein portion of the enzyme either covalently or non-covalently. Some cofactors are tightly associated with the protein covalently or non-covalently, or called prosthetic group. As for example, vitamin B complex and their coenzymes, thymine, that is B1, and the coenzyme form is thymine pyrophosphatase. Riboflavin, that is B2, contains FAD and FNM cofactor. Pyridoxine B6, that contains pyridoxal phosphate cofactor. Nicotinic acid niacin, NAD plus and NADP plus coenzyme form. Likely pantothenic acid, that is B5, contains coenzyme A. Biotin contains biocytin cofactor. Similarly, folic acid contains tetrahydrofolic acid cofactor, and vitamin B12 deoxyadenosylcobalamin cofactor. Naming and classification of enzyme. Many enzymes have common names. For example, trypsin, a proteolytic enzyme, is secreted by the pancreas. Common name provides little information about the reaction that enzymes catalyzes. Many enzymes are named for their substrate and for the reaction that they catalyze, with the suffix is added. As for example, ATPase is an enzyme that help in the breaking down ATP, whereas ATP synthesis is an enzyme that helps in the synthesis of ATP. Because of the confusion that arose from these common names, an international commission on enzyme was established to create a systematic basis for enzyme nomenclature. The Enzyme Commission has developed a rule for naming enzymes. According to the rule, each enzyme is classified and named according to the type of chemical reaction it catalyzes. Enzyme Commission, that is EC, has given its name according to the type of chemical reaction it catalyzes. Enzymes a number with 4 parts like EC2712, that is hexokinase. The first 3 numbers define major class, subclass, and subclass respectively. The last number is serial number in the subclass, indicating the order in which each enzyme is added to the list. As for common name and EC number of some enzymes, alcohol dehydrogenase EC1111. Similarly, phosphoproctokinase EC is 271111. Systematic classification. The first integer in the EC number designates the class of enzyme. There are 6 classes to which different enzymes belong. These classes are oxidoreductase, transferases, hydrolases, and lysases, isomerases, ligases. First of all, oxidoreductase. Oxidoreductases catalyzes oxidation reduction reaction. Some examples are oxidases, dehydrogenases, oxygenases, and peroxidases. The function of oxidases is use oxygen as an electron acceptor but do not incorporate into the substrate. Dehydrogenases use molecule other than oxygen, e.g. NAD+ as an electron acceptor. Oxygenases directly incorporate oxygen into the substrate, and peroxidases use H2O2 as an electron acceptor. Second enzyme is transferases. Transferases catalyzes reaction that involves the transfer of groups from one molecule to another. Example of such group include amino, carboxyl, carbonyl, methyl, phosphoryl, and acyl Common trivial name for the transferase often include the prefix trans. As for example, transcarboxylases transfer say carboxylate group to substrate. Transaminases transfer amino group from amino acid to keto acid. Kinases transfer phosphate from ATP to substrate. Similarly, phosphorylases transfer inorganic phosphate to substrate. The third enzyme is hydrolases. Hydrolases catalyzes reaction in which the cleavage of bonds is accomplished by adding water. Some examples are phosphodiesterase, which cleave phosphodiester bond. Phosphatases remove phosphate from substrate. Peptidases cleave amide bonds such as those in proteins. Now, lases. Lases are enzymes that catalyzes the breaking of CC, CO, CN, and CS bonds by means of their other than hydrolysis or oxidation. These bonds are cleaved by the process of elimination and result in the formation of a double bond or a new ring, or conversely adding groups to double bonds. Some examples are aldolases, removal of aldehyde via elimination reaction; synthesases, linked to molecule without involvement of ATP; dehydratases, removal of H2O via elimination reaction; and decarboxylases, removal of CO2 via elimination reaction. And the fifth enzyme is isomerases. Here, isomerases catalyzes several types of intramolecular rearrangements and yield isomeric forms. Some examples are mutases, catalyzes the intramolecular transfer of functional groups. Cis-trans isomerase, it catalyzes the isomerization of geometric isomers. Epimerases catalyze the inversion of asymmetric carbon form, and racemases interconvert L and D stereoisomers. The last enzyme type is ligases. Ligases catalyze the formation of CC, CS, CO, and CN bonds. The energy for this reaction is always supplied by ATP hydrolysis. Other common name for ligases include synthetase because they are used to synthesize new molecules. Example, carboxylases use CO2 as substrate. So, students, these are the general properties and classification of enzymes. Thank you very much.